Hello, in this video we will discuss about the mTOR pathway and their significance in proliferation of cells. So how to react this mTOR pathway and mTOR complex 1 and 2. So important thing is that first of all we should need to understand the mesosome which that is present in the cell here. So here is the mitochondria, nucleus, and rough endoplasmic reticulum, as well as there are several organelle showing in the cell. Here is the Golgi bodies. So in this way you can see these Golgi bodies, the mitochondria, which that is a powerhouse of the cell, and this is the nucleus, which that contains genetic material inside and control the genotype. And on the other hand, here is the mesosome or present endoplasmic reticulum, and here we will just understand about the mesosome for the autophagy. So important thing is that let's begin to understand that here is the food vacuole, for example extracellularly present, which that is combined together and packing, and that is the endocytosis will be occur. Endocytosis means the endo mean inside, cytosis means the movement of into the cell. So it can be pinocytosis or phagocytosis. Phagocytosis is the solid while the pino is the liquid. Let's begin to understand the mechanism, and here is then go into the endocytose the food particle, and this go and in the form of vesicle, and it go into the mesosome. While here is just we will understand about the intracellularly organelle, for example mitochondria, which that will be damaged, and phagophore formation, and this is the autophagosome formation, and this autophagosome will be bind with the mesosome by a different reaction mechanism to fusion to endocytosed, move engulf the it is can be pino and phago. So the pinocytosis or phagocytosis. So in this way there are different autophagy, which that is known as the autophagy. It means the macroautophagy, microautophagy, crinophagy, and chaperone mediated autophagy, and mitoautophagy, which that is the mitochondria use, and that content will be moved inside into the mesosome. So the hydrolytic enzymes use phosphatase, etc. This used to degradation. Degradation after this content will efflux through a channel, and in this way this can be recycled as recycled matter, and cell repair using for cell repair and organelle formation. So in this way here is you can see this is the mesosome, which that is important for the recycling of the content in the cell, intracellular damaged content. So damaged content will be recycled for the prevention of the proliferation and differentiation of the cell, as well as the genetic material changes, because the mitochondria contain own genes, and when gene will be differentiate or mutate, so that will be engulfed to form a phagophore. So it's the mTORC1 complex, which that is contain daptor, MLSTs, and raptor, and pras40, which that is stimulate and inhibit the 4EBD1, which that is used for the protein synthesis inhibition. By this protein synthesis will be occur, and S6K1, which that is used for the nucleotide synthesis and protein synthesis, while here is the SREB-P1 is the used for the SREB-P1 cleavage formation to increase the lipid synthesis, and CYT4 which that is the expression to increase the expression to glutamine metabolism, and H1F1 alpha C mice, which that is used for the glycolysis, while the autophagy will be inhibited by this mTOR complex. Why autophagy will be inhibited? Because this is going to the synthesis pathway. So remember about that. When synthesis will be synthesis pathway will inhibit the autophagy by proliferation, and more growth will be occur in the cell. So let's begin to understand during the eating time. So the nutrient rich condition when we will eat, or in our body will be more nutrients. For example, sodium, amino acid and glucose and fat. So in this way the diabetic person can be increase the glucose level also, and amino acid by the proteolysis. So in this way, here is the amino acid will be moved inside through extracellular to intracellular, use ATP to potassium and sodium exchange. Here you can see, and the sodium will move inside with one amino acid, as well as GLUT4 channel is used for the glucose movement, entry of the glucose. After the movement in the glucose, the glycolysis will be occur to increase the ATP with the ratio of AMP. And let's here is the nutrient rich condition, while here is I'm drawing the starvation condition. Starvation condition mean we are not eating and we are going to starvation or fasting stage. So during the nutrient rich condition, here is the activated mitogen protein kinase, which that it will be activated with the LKB1. After this LKB1 stimulation, so it can inhibit through increase of the ATP into the cell. So during that rich condition, nutrient rich condition, it will be inhibit. While here is the tyrosine kinase receptor is attached with the growth factor, for example insulin and growth hormone, to phosphorylation, and this phosphorylation will be growth receptor bound protein 2 with bind with the source and activate the RAS GDP to convert the GTP, RAS GTP. After this conversion, the RAF, this is the soluble RAF. Soluble RAF means the rapidly accelerated sarcoma protein is the soluble, and the soluble protein can be activated with the phosphorylation with a membrane bound RAF. With a membrane bound RAF that will be phosphorylation, and this mitogen activated protein kinase, which that in the presence of their substrate, their activation. So this membrane bound RAF will activate the mitogen activated protein kinase inactive to become activated. So the mitogen activated protein kinase activation with the phosphorylation, and to convert the MAP kinase mitogen activated protein 2 kinase will remain. And here is the arc. This is the arc, basically is the inactive to activate, and MAP kinase will remain, and the arc will phosphorylate. After this arc phosphorylation, it used for the transcription of the different gene we will discuss later. But here is the mTORC and mTORC2, which that in enhance the MAP kinase, arc protein. So the arc phosphorylation will be increased through mTOR to mTOR pathway. So here is the phosphoinositide 3 kinase will convert, stimulate the AKT to inhibit the TSC1 and 2 to inhibit the RHEB with GTP formation. So in this way, the inhibition of this will stimulate more mTOR pathway, while the rapamycin can be inhibit the mTOR pathway, mTOR complex and mTOR pathway. So this is the mTOR pathway I am drawing here with the lysosome. So here is the mTOR pathway will triggered by the phosphorylation of the RAP, GTP and mTORC1 complex, which that MLSTS and Raptor and PROS40, which that also phosphorylase the RAG A and B GTP conversion, and RAG C and D dephosphorylase to GTP to GDP. And this complex, and here is the regulator, this complex will inhibit the sodium move out, so the efflux of the sodium will inhibit. So here is the regulator. This regulator means the proton pump will be produced, and if in this way the acidification will be decreased inside of the inside of the mesosome. So the movement of the proton will create the ATP, and on the other hand the microtubules will be formed and anterograde transport. So on the other hand, here is the nucleus. So arc will be, again I am talking about the arc is used for the transcription. So here the transcription factor will bind by the stimulation of the arc. So arc will stimulate the transcription, by the transcription factor and a phosphorylation to transcription. After this transcription, the autophagic and lysosomal genes autophagic and lysosomal genes will be basically will be inhibit those. So without arc, basically here is you can see this will during the starvation time, while here is the nucleus, which that is the autophagic and lysosomal gene will be inhibit because arc will not bind within it. So important thing is that the arc will not bind. So important things that you should need to understand. So in this way, here is the transcription factor, which that is basically transcribe. And while in this way, you can understand as well the autophagic and lysosomal gene will be inhibited by this, and the transcription factor will proliferate and synthesize the protein. While here is the GDP, RAG AB, and RAG CD, which that is activated. And here is the TF transcription factor EB and transcription factor E3, which that is mTOR C1, which activate. After this activation due to the separation, so in this way you can see, so that mTOR C1 when remove, remember about that, we remove. So in this way it will be stimulate after the starvation condition. So it will phosphorylase through ADP to ATP dephosphorylation. So after dephosphorylation, the autophagic and lysosomal gene will be activated. The transcription factor EB and transcription factor E3, which that stimulate the ZK-scan3 protein genes activation. So the transcription factor, which that translate and produce the protein. And here you can see the microtubules increase during the nutrient rich condition to increase the proliferation of the cell, while the lysosomal biogenesis will be occur. And in this way the increase of the autophagic activation during the starvation mode. So important thing is that here you can see when detached this this RAG A, B, and CD. After the detachment, the transcription factor EB and E3 will be produced. This E3 and will be transcription factor will stimulate to produce the autophagic activation, and in this way the lysosomal biogenesis will be occur due to this after the detachment. While when will not detach, the autophagic and lysosomal gene will be not produced, as well as the ZK scan 3 transcription of this gene will not promote. So in this way important thing is that here is the autophagophore will be formed. After this, due to the nutrient will be attached, and if a autophagosome formation, this autophagosome will be fused. After the fusion, so the here is the autophagosome, that starvation condition, and in this way here is the lysosomal activity will be increased. In this way the activity of the lysosome, because the proliferation of lysosome will be more, and acidification will be increased, and the amino acid level will be decreased, and acid is proton will be increased, so the efflux of the sodium will be increased, and depolarization will be increased, and in this way the ATP sensitive sodium channel will be enhanced. While here you can see the autolysis or reformation will be occur. So the reformation mean the mTOR C1, which that will be inhibit the autolysis, while the mTOR C1 will be promote the reformation. So it will maintain the lysosome and not degradation and not recycling, while the mTOR C1 will be inhibit the inhibition of autolysis of the mesosome, because the autolysis will uh, will go to the cell apoptosis. So it will inhibit the apoptosis, while mTOR C1 will promote the proliferation to lead the cancer cell. So the cancer causing mTOR complex is resembling with the cancers and proliferation of the cell. On the other hand, important thing is that you should need to understand that during the nutrient rich condition, the mTOR C1 activation, while the inactivation during the starvation condition. So thanks for watching. Please make sure to subscribe.